One-dimensional 1D electrophoresis is a widely used technique in molecular biology and biochemistry for separating molecules, particularly proteins based on their size, charge, and other properties. It works by applying an electric field to a sample, which drives a charged molecule through a gel matrix. Principle of 1D electrophoresis. First, electric field. An electric field is created by applying a voltage across the gels, and this causes charged molecules, protein and nucleic acid to move toward the electrode with the opposite charge. For protein, this is typically the cathode negative or anode positive, depending on the molecule net charge. Second, size. The geometric act as a sieve, allowing smaller molecules to move through it more easily and rapidly than larger molecules. Larger molecules experience more resistance as they travel through the gel, leading to slower movement. These size-based separations allow for the sorting of molecules by their molecular weight. Third, charge. Proteins or other molecules are charged positive or negative depending on their amino acid compositions or structure. This charge influences how fast and in which direction they move in the electric fields. The movement of molecules is governed by the principle that oppositely charged particles will move toward the electrode with the opposite charge. Cation move toward the negative electrode, and ion toward the positive electrode. Fourth, shape. The shape of a molecule can impact its migration speed through the gel. A globular protein, for example, may migrate differently compared to a highly elongated protein. The more compact a molecule is, the more efficiently it might move through the gel compared to a more irregularly shaped one. 5. Medium. The medium used for the electrophoresis, usually a gel such as agarose or polyacrylamide, affect the separation process. Different gels provide different level of resistance to molecule movement, and the concentration of the gel can affect how well it separates molecules of different size. For example, a denser gel higher percentage of polyacrylamide is better suited for separating smaller protein. Applications of 1D electrophoresis. First, protein separation. One of the most common use of 1 electrophoresis is the separation of protein based on their molecular weight. By comparing the migration distances of proteins in the gels to a set of known molecular weight standards, the size of an unknown protein can be determined. Second, protein purifications. During the process of purifying proteins, 1D electrophoresis can be used as an analytical tool to monitor the progress of purification steps. By analyzing the bands on the gel, researchers can determine how pure their sample is and whether there are contaminants or multiple forms of the proteins. Third, sample purity. 1D electrophoresis is a valuable tool for assessing the purity of biological samples by running a samples on the gels and checking for the number of band present. One can quickly see if the sample contains a single protein or multiple protein. If multiple bands appear, it suggests that the sample is impure. Fourth, molecular weight estimations. For proteins whose molecular weight is not known, 1D electrophoresis provides a way to estimate it by comparing the positions of the proteins of interest relative to a molecular weight ladder, a set of known proteins of different size, and approximate molecular weight can be deduced. Operation. First, prepare the sample. Weight samples and dissolve them in sodium dodecyl sulfate SDS polyacrylamide gel electrophoresis. Sample to be run on SDS paste are firstly boiled for 5 minutes in sample buffer containing B-mercaptanol and SDS. Second, prepare gel and buffer. Dissolve gel in boiling water, put it into a mold and create wells in the gel with a comb. Third, load and pipette sample. Load the sample into the wells in the gel. Fourth, run the gel. Apply an electric current to move the protein through the gel. 5. Visualize and document bands. Use molecular weight standards to determine the size of the protein. The mercaptanol reduces any disulfide bridge present that are holding together the protein tertiary structure, and the SDS binds strongly to and denature the protein. Each protein in the mixture is therefore fully denatured by this treatment and opens up into a rod-shaped structure with a series of negatively charged SDS molecules along the polypeptide chain. On average, one SDS molecule binds for every 2 amino acid residue. The original native charge on the molecules is therefore completely swamped by the negatively charged SDS molecules. The sample buffer also contains an ions able striking dyes, usually bromopenol blue, that allow the electrophoretic run to be monitored, and sucrose or glycerol, which give the sample solution density, thus allowing the sample to settle easily through the electrophoresis buffer to the bottom when injected into the loading well. Once the samples are all loaded, a current is passed through the gel. The sample to be separated are not in fact loaded directly into the main separating gel. When the main separating gel, normally about 5 cm long, has been poured between the glass plate and allowed to set, a shorter approximately 0.8 cm stacking gel is poured on top of the separating gels, and it is into this gel that the wells are formed and the proteins loaded. The purpose of this stacking gel is to concentrate the protein sample into a sharp band before it enters the main separating gel. This is achieved by utilizing differences in ionic strength and pH between the electrophoresis buffer and the stacking gel buffer, and involve a phenomenon known as isotopic.
Advantages of 1D electrophoresis. Versatility. It applies to various types of binary molecules. For example, SDS pitch, determining the molecular weight of purified protein. Native pitch, investigating protein-protein interaction or enzyme activity in functional protein complex. Agarose electrophoresis is applied for assessing the quality or size of DNA and RNA fragments after PCR. Then, it's cost-effective because 1D electrophoresis is not as expensive compared to more advanced techniques such as 2D electrophoresis or mass spectrometry. It relies on common laboratory supplies such as gels and buffers. Third, it 1D electrophoresis also has high resolution. For example, isoelectric focusing can achieve extremely high resolution within the isoelectric point range of proteins, suitable for separating isoform and minor charge differences. Quantification and visualization. For example, SDS page and native page are compatible with various staining techniques such as chromatography, silver staining, and fluorescent dyes to detect and quantify separated molecules, making them versatile technique for protein visualization. Limitation of 1D electrophoresis. 1. Lack of complexity analysis. For example, SDSPH cannot resolve if protein sample contains isoform with different post-translational modification, such as glycosylation, and need 2D electrophoresis or mass spectrometry for detailed analysis. Second, post-processing requirements. 1D electrophoresis require additional steps after separation, such as agarose, gel electrophoresis, fragment size estimation, or further purification of separated components are required. In isoelectric focusing, transfer to secondary techniques such as 2D page or immunodetection for downstream analysis is required. And third, susceptible to contamination. When the electrophoresis is susceptible to contamination, which can compromise the result. For example, contaminated samples. Impurities in biological samples can cause smearing or distorted bands. So it will impact and interfere with protein or nucleic acid separation. Then impurities in buffers, such as with salt, organic material, or microorganism, can affect migration patterns or conductivity. Apparatus contamination also can produce smears, streaks, or irregular migration patterns. Fourth, one electrophoresis also has time limitation because it's time consuming and labor intensive. Each technique involved in 1D electrophoresis requires several steps that needs careful optimization, such as gel preparation, sample loading, running the gel, staining, destaining, and imaging. There are several examples of real-world application of 1D electrophoresis. First, uh, for the study conducted in 2020 titled Agar's Native Gel Electrophoresis for Characterization of Antibodies shows that native agar gel electrophoresis in zero point 0.1 molar His zero point 0.1 molar mass or zero point 0.2 molar buffer can be used to distinguish different monoclonal antibodies and evaluate their purity. The agarose gel electrophoresis can also be used to see a protein profile in native state of varicella and detect aggregation or degradation. Second, in the case study titled TCA precipitation ethanol/HCl single step purification evaluation, one-dimensional gel electrophoresis, Bradford assays, spectrophotometry, and Raman spectroscopy data on HSA arenases Lysozyme basket skyline data. In this study, conducted in 2018, one desired electrophoresis was used to analyze the protein profiles of various samples, such as mouse liver, river biofilm, and loamy soil surface. The technique helped in separating protein based on their molecular weight, allowing for the evaluation of protein recovery and purification efficiency. A third case study was conducted in 2019. This is interesting because it was conducted relating to the Malayan pit viper. The study, titled Comparative Proteomes, Immunoactivities and Neutralization of Procoagulant Activities of Cholestylasma rhodostoma, Malayan pit viper, venomous from four regions in Southeast Asia. In this study, 1D SDS was used to analyze the venom proteins of Ceratostoma Malayan pit viper from different regions in Southeast Asia. The technique helped in separating the venom protein based on their molecular weight. As a conclusion, 1D electrophoresis remains a critical tool in analytical biochemistry due to its versatility, simplicity, and effectiveness in biomolecular analysis. From protein separation to gene identification, its application are vast and central to research, clinical diagnostic, and industrial biotechnology. Despite several limitations, it offers valuable insight into the structures, function, and integrity of biological molecules. Continued and future advancement ensure that one, the electrophoresis remains one of fundamental analytical technique in biochemistry. Thank you.